Hello, we're Team Lad, and today we'll be presenting our study conducted throughout the semester elucidating the function of the Bradi1g72430 gene in the plant Brachypodium distachyon. Our two main research goals of this investigation are to first identify the function of the gene, and secondly, identify the unknown mutant and effect of that mutation. From these findings, we hope to be able to develop a better understanding of the overall gene function and the plant as a whole, which could possibly be utilized in applications such as recombinant technology, synthetic polymer biology, or perhaps even health, depending on the function of the gene.
So this past semester, between January and May of 2020, we performed extensive research to better understand the Bradi1g72430 gene. Things include understanding the location of the unknown mutation from GeneMap and bioinformatics, structure and function through literature searches, understanding orthologs through phylogenetics, presence of mutation in plants from DNA extraction, gel electrophoresis, PCR, and Sanger sequencing, mutation on the phenotype from cross-section analysis, and regulation of the gene itself through data mining and RNA-seq analysis.
Using bioinformatics, we constructed computer-based models and were able to develop a holistic map of our gene. So the first ab initio model was constructed using F-Ganesh, and the output revealed a result of 15 predicted exons, which the RNA-seq models also predicted as well. The homology-based model predicted 11 contiguous sequences, a mixture of high and medium confidence, but based on visual observations, they're also mostly aligned with the F-Ganesh and RNA-seq model outputs. We predict that it's probably a fewer number of contigs than the 15 exons predicted, since if we look at the red lines on the homology-based model, some of them span more than one exon region. Overall, the four models predict a very similar structure of our gene. Analyzing the gene on the phytosome site, we found that the mutation was located at the site NAN1898 as indicated by the red diamond, and was also marked as a high impact mutation. However, we saw that the mutation was located in the 5' untranslated region of the gene. Due to its location, we thought that it would have likely no effect on the phenotype.
The NCBI protein blast output yielded two conserved domains, leucine-rich repeat and receptor-like kinase domains. Together they form an LRR-RLK domain, which are often found in a superfamily of proteins. The LRR-RLK protein consists of two domains that sit across a bilipid membrane. The LRR domain is on one side, whereas the RLK domain sits across the membrane itself. These two proteins act as co-receptors, and they heterodimerize to transduce a signal via a MAPK cascade and results in gene expression. This gene expression is categorized into two groups: growth and development and defense-related responses. Growth and development includes overall plant growth, stomatal development, root development, xylem differentiation, and male gametophyte development. And responses and related to defenses includes floral abscission and immune response. Our gene of interest was likely involved in some way or another with the LRR-RLK function and signaling pathways, and this conserved domain was used as a base for literature searches in order to understand the functions of these domains across various species. There are a bunch of known LRR-RLK genes that have known functions to play a role in cell growth and development. Specifically in Arabidopsis, there is the PXY-TDR correlated genes, and they are known to reduce secondary cell wall development. So looking at three different mutations of this gene, mutations 1 and 2 are in the coding region, and mutation 2 is in the UTR, similar to our mutation, except ours is in the 5' and theirs is on the 3' end. We can see that there is a reduced expression of secondary cell wall development in the mutant plants compared to the wild type, and the plants themselves are droopy and cannot support their own weight after 47 days.
Protein blasts of the gene of interest also revealed a high degree of homology with the class of transmembrane receptors called strubilig, which are known to play a role in mediating tissue morphogenesis as well as the cell wall damage response. In a study conducted by Chowdhury and colleagues, they decided to test the effect of isozabin treatment on lignin and callose biosynthesis on wild-type and mutant plants in Arabidopsis thaliana. Isozabin is an herbicide that inhibits cellulose biosynthesis by targeting the cellulose synthase complex on the plasma membrane. So in the first study, lignin accumulation is detected on plants treated and not treated with isozabin. The wild-type strands are shown as LER and COL, and their mutant counterparts are shown to the right. Differences in staining between the wild-type strands and their mutant counterparts are shown with a decrease of lignin accumulation on the mutant plants. This is also shown in the graph in panel B. In the next experiment, callus formation was detected in response to isozabin treatment, comparing the wild-type plant COL and its mutant variant SUB9 in panel C. Upon isozabin treatment, you can see a higher amount of callus detection in the wild-type plant in comparison to its mutant. This is also shown in the graph in panel D, where the wild-type plants have a higher amount of callus accumulation when compared to their mutant counterparts. Overall, these findings suggest that proper strubillig function is needed for lignin and callus formation in response to cell stress. So in order to better understand the function of the gene, evolutionary relationships of the Bradi1g72430 gene with other plant species was analyzed by building a phylogeny. So using the data obtained from the NCBI protein BLAST tool, genes closely related to the gene of interest were identified in the following 5 plant species indicated in the key. So based on the tree, the most closely related ortholog was found to be in Oryza sativa, a rice plant, where our gene is indicated in the path highlighted in blue. 2 other orthologs were also identified in Arabidopsis thaliana, indicated with the 5 and 6 below the rice ortholog. During this process, the protein BLAST also revealed a 100% identity among the protein strubillig receptor family 7, supporting our hypothesis of the gene encoding for some protein in the strubillig class of receptors. We sent 4 of our plant samples off for sequencing, 1 wild type and 3 mutants. Shown on this slide are the Sanger sequencing results from PCR. Mutants 3 and mutant 6 were identified to be heterozygous mutants with the presence of G and A at the mutation locus, G being the quote-unquote wild-type allele. Wild-type 1 was found to be homozygous wild-type. However, mutant 4 was also found to be homozygous wild-type as well, and not actually, in fact, a mutant as initially thought. So these results indicate that the parental plants were likely heterozygous due to the complete absence of any homozygous mutant plant, which would have the chromatogram result shown in panel B. However, a larger sample would need to be sequenced in order to be totally sure.
We took 10 measurements of interfascicular fibers for 4 different cross-sections of both the wild type and the mutant plants, and we found that there was no significant difference between cell wall thicknesses between the wild type and the mutants. We repeated this process for epidermal cells by measuring the outward-facing side of the epidermal cell, and we took 10 measurements for 4 cross-sections of both the wild type and the mutant plants, and again, we see no significant differences between cell wall thickness between the wild type and the mutant plants.
The expression of the Bradi1g72430 gene over time in varying light and temperature conditions was analyzed using data published by Dr. Hazen in his lab, along with the experimental data from the EFP browser site. So based on the results obtained, the gene is likely not regulated by thermocycles or photocycles as shown in the top graph, where there is no discernible regular cyclic movement in any of the 4 light, dark, and heat conditions. However, as indicated in the heat map below under constant light and heat conditions, the gene is possibly regulated by circadian rhythm, as the RNA-seq analysis results showed a higher expression of the gene about every 14 hours, indicated by the green arrows.
Results from an EFP browser revealed that the gene was most highly expressed in stem, root, and leaf tissues, and more specifically, flag leaf, mature leaf, and young leaf tissues, which is what we will be focusing on. Looking at the stem, leaf, and root tissues, we do see a significant difference between all three of them, as the p-value was less than 0.017, which includes the Bonferroni corrected p-value. We see the lowest expression in the leaf tissues and the highest expression in the stem tissues. Now, looking at the young leaf versus the mature and the flag leaf, we do see a significant difference in expression between the young leaf versus the mature leaf and flag leaf, but no significant difference between the flag leaf and the mature leaf, and the flag leaf and the young leaf itself.
Using a pre-made count matrix on expression levels of various B distachyon genes based on RNA-seq data across the selected conditions of wild-type overexpressing mutant and mutant 6, a plot of expression levels of the gene was generated for the gene of interest using RStudio. So based on the results, the highest level of gene expression was observed in the overexpressing condition as expected, with mutant 6 having a slightly lower level of expression of the gene compared to the wild-type condition. Although this analysis was performed on data we did not collect in our own studies, this activity demonstrates the application of using a tool like RStudio to conduct RNA-seq data analysis and a technique to consider for future experiments.
So in summary, the Bradi1g72430 gene is likely a Strublig receptor family 7 gene. It contains an LRR-RLK, and a protein blast revealed 100% homology with this class of receptors and close matches of other families of Strubligs. It's likely to mediate response to cell wall damage, and cross-section analysis confirms that a mutation in the UTR has no effect on phenotype. Its expression is greatest in the stem tissue, and benefits of these findings can help us better understand the gene function as well as the organism as a whole, and this can be used in applications in recombinant studies or technology and can potentially help us understand that mutations in the coding region of the gene through knockout studies may have phenotypic impacts. Repeating studies across orthologs may help us understand the conservation of this gene function across many different species as well. A huge thanks to Professors Sam Hazen, Katie Webster, and Catherine Dorfman for helping us out with all this research, and we would not have been able to conduct this study without you, so thank you so much.